Quiet. That's right. Well, let's go ahead and get started.
Anybody else on the way?
Terry will be coming for it. I think everybody knows Dr. Al Johnson. He is, as you can see, he's a distinguished professor of radiology, a professor of physics and biomedical engineering, which just like blows my mind. But anyway, he is here to tell us about some of his work today.
Thank you.
And I will let you take it from there, because I don't really understand it. So that's why I won't.
The thing is, I don't either. It's fooling some of the people some of the time, or some of the people, the right people at the right time. That's right. So, please interrupt me. Please ask a question. It will make it so much more fun for me and you. So, I'm first an apology for those of you who were expecting to hear about clinical MRI. That was an earlier part of my life. In the first, from 1974 through 2009, I was the director of physics for the Duke Healthcare Center, and I was involved in really useful stuff like putting CT and MRI systems in. I left that world to start doing, yes, sir.
I'm hard of hearing,
I know that.
so I can barely hear it.
I will be much louder. I'm hard of hearing as well. Okay, so I'm going to talk a little bit about an adaptation of MR to do basic research. So just to give you an orientation, this is an image of the mouse brain. It's a slice of the mouse brain. And what you're looking at is not MR. The blue is actually a type of optical microscopy. What's superimposed on it are tracks that are calculated and generated from MRI. And these tracks here are acquired at spatial resolution that's about 35 million times higher than what you would get in a clinical setting. So have I wasted my life? Have I wasted my life? I'm going to try to show you that I didn't waste my life. I'm going to say a little bit about just an introduction to what MRI is. I'm not going to spend time telling you how it works. I'm going to spend most of the time talking about MR microscopy. Can you see okay, Lisa?
Yeah, I can speak a little one.
And then finally, I'm going to try to reduce it to something that is digestible, how we're using it to understand Alzheimer's disease, certainly something close to my brain. So, 1973 was a very good year. A very good year because the Nobel Prize for CT, the 1979 Nobel Prize for CT, the work was published by Alan Cormack, a mathematician, and Godfrey Hounsfield. Godfrey was an engineer who worked for the Beatles company, ENI, the record company. And in that same year, these two publications from Paul Lauterbur and Peter Mansfield came to fruition. Those became the basis of the Nobel Prize in Physics and Medicine for 2003. CT caught on earlier, quicker. It was an easier technology to master. And the thing that CT brought to the party was transverse axial scanning. Up to this point, when you went to get an x-ray, you put your hand in front of a film, x-rays came in, you got a projection of your entire hand. You stood in front of a cassette, x-rays came in, you got a chest film, but you didn't get a slice. And what CT did was said, oh, I can give you a single thin slice. So suddenly transverse slices were possible. Yes, sir. I'm always intimidated by Oxford t-shirts. Go ahead.
Is CT computerized transverse?
Yes.
Okay.
So, and you'll see that it's transverse because here's a CT, we'll go through this. This is a typical CT scan. What's the big deal about MR? Well, CT is showing you something about the X-ray density. So, bone, the density is very high, so it's very bright in a CT scan. These are the ventricles, and you don't see a lot of difference in the soft tissue detail here. There's gray matter, the stuff that does the thinking, and white matter, the stuff that connects the stuff that does the thinking. This is the MR scan of the same patient. So, does this help? Testing 1, 2, 3. 1, 2.
Keep shouting to it.
1, 2, 1, 2.
There we go.
Jeremiah was
Great bird.
So in the MR scan, the MR is showing you something completely different. It's showing you soft tissue contrast. These are the ventricles filled with cerebral spinal fluid. You don't see any signal from bone. Bone has no protons. MRI is looking only at signal from protons. So you don't see the bone. You see a little bit of signal where you might think there's bone, but that's just fat in the marrow inside the bone. Yes, ma'am?
For a layperson, what makes the difference in the densities that they show up?
I'm about to tell you that. So let's go with what CT does first. CT starts with an X-ray tube above you, a detector below you. The detector has on the order of 1,000 different elements measuring all the X-rays propagating through. This rotates around you and takes projections here, 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 all the way around, and then projects them backwards to create that slice. And what it's projecting is the density of X-ray opaque material. So you're just looking at X-ray density. MR, to your question, is looking at protons interacting with the magnetic field. If you look at a proton at a quantum mechanical level, at a microscopic level, it's a little charged particle. It's spinning really, really fast. And as a consequence, it looks like a gyroscope. So you put a gyroscope on your finger, gravity will tend to pull it over, and it will precess. The same thing happens with MR. That little magnet, once placed into a magnetic field, will begin to precess just like the gyroscope. So what that does for you is a collection of protons. Your wrist, your head, your leg, whatever, is a collection of protons. You put them in a strong magnetic field, and enough of them will start precessing synchronously with the same frequency so that you get a magnetization, which is something you can measure. With me so far? And we'll get to the better question in a moment. You can probe this by putting another magnetic field in there. That's the magnetic field from a radio wave. Radio waves are oscillating electric and magnetic fields. So at 1.5 Tesla, which is where 1.5 to 3 Tesla are where most of the clinical systems work, you're running something between 60 and 120 megahertz. That's sort of around WQDR. So you're transmitting radio waves into the patient, and you're getting a radio wave out. And the radio waves in do the excitation, and you then encode a whole bunch of different things about how that proton is interacting with its environment. So microscopy, we'll come to a little more in another 2 slides. Microscopy was what Paul Lauterbur thought he could get happening. This paper in 1973 concluded, the very last line was that you could look at, he talked about zoidmatographic techniques. By that he meant imaging techniques. He said imaging could be done at microscopic resolution. So Paul understood that back in 1973. It took a long time to get to the point where you could do it, but he knew that you could. And this is the very first MRI system, the very first MRI images. Parenthetically, I had the opportunity to go to the University of Illinois about 4 or 5 years ago. They wanted to do, they wanted some people with clipboards, experts, coming to tell them. So I was one of those people. We were standing in the hall, in the basement, and there was a bunch of Paul's crap from his laboratory. It had been put in a shed. University of Illinois is a farm university. It's right at the edge of Champaign. And it was out literally in a farm shed, all of this stuff. And there was a strip of paper that had one of these traces. What he did, he had a magnet like was used by chemists. Paul was a chemist, and he put 2 capillaries in there. And he changed the magnetic field so that as you went from here to here, or from here to here, as you go across, the magnetic field changed. The direction remained the same, but its strength changed. So as the strength changed, the precessional frequency changed. So the frequency here was different than the frequency here. So he just took a spectrum. He said, give me all the signal. And all the signal coming from this one shows up there. All the signal coming from this one shows up there. So it looks a lot like what you were getting from the projection of an X-ray system, except now it's the proton signal. But what can you do with that proton signal? Well, this is where we're getting into the meat of what I'm interested in. We talked about doing magnetic resonance histology because in 1981, my chair, who lived over on Mount Sinai Road, called me one night about 9:30, said, Al, are you busy? The chairman of radiology calls you at 9:30. And I said, no, sir. So I hustled over to Charles' house. Charles was a very dynamic individual. He was a Texan. The most interesting thing about Charles was he could spit with precision. He had a tune, and he would carry on a conversation and he could spit. It was just so impressive to see. And we talked about, we were doing CT at the time. We had certificate of need for 2 and we had 7 CT scanners. So we were illegal by a factor of 5. And I thought that's what we were going to talk about. We said, no, we're going to talk about putting the biggest goddamn magnet in the world at Duke. And he had struck a deal with another Texan at GE to put the very first high-field MRI system in the world at Duke. So from 1981 to on through 2009, my job was to see that that worked. And I said, oh my goodness. We at the time could do really good CT scans. Why were we doing this? Well, it'll become obvious in a minute why that would be useful. But the first studies that we were doing, I was doing in the hospital with small animals, mice, rats. And the problem for an engineer is when the rat wakes up in the middle of the night, crawls out of the magnet and goes out into the hospital someplace. It's underneath the computer floor in someplace and you've lost all your signal. From an engineering standpoint, the signal has just disappeared. It's down the corridor someplace. So we got interested in looking at tissue only because it didn't move. And so that brought us up to magnetic resonance histology. I'm going to show you why that's still useful. And it's useful because we get proton stains. I'll show you a proton stain in a moment. It's non-destructive. We don't slice or dice anything. We get a chunk of tissue, we image it, and we can do other things with it. We can do molecular biology. We can do conventional optical scans. It's 3D. Back in 1993, having a 3D scan was a really big deal. Now it's not a big deal, but in 1993 it was a big deal. And then finally, it's inherently digital. Again, in 1993, that was not a big deal. One of the grants that I got, I think about 1990, 89, was to link a whole bunch of MR and CT scanners. Yes, we had the MR and CT scanners together. I got a perfect score, the only time I've ever got a perfect score, and it was to do something called networking. So I am the Al Gore of Duke. So we're talking about proton scans. You asked why. So here is MRI of a patient with a stroke. This is what's called a T1 weighted image. You excite the tissue and you wait, and you excite the tissue again and you get a signal. You excite the tissue again, you get a signal. Every time you excite the tissue, if you were to excite immediately after that, you will get no signal because you've already excited it. If you wait 100 milliseconds, 200 milliseconds, 300 milliseconds, you'll get more and more signal, depending upon how rapidly the first signal that you excited decays and translates into the tissue around. So this is fat. Fat has a very short relaxation time. That is, you stimulate it and you can stimulate it again really quickly. So if you're pulsing rapidly, you'll get a lot of signal from fat because it's recovering quickly every time. You get very little signal from the CSF because it's pure water. Water has a T1, a relaxation time of about 3 seconds. you'll see some differentiate. These are very old scans now, so they're pretty crappy, but they show the point. You see some differences between gray and white matter. If you're a clever biomedical engineer, these things will not hurt you. They like to have fun, so you must name them T2 and T1. And so we had to have another parameter. You must know that story. Do you know the entire Cat in the Hat?
Not the entire thing, but I do know the Cat in the Hat, yes.
I know a lot of it. It was somehow pretty deep in my brain. So here's the T2. You excite the tissue, and the signal rings for a long time. And the more rapidly it decays, the shorter its ring time, the shorter the T2. So water that had a very long T1 and was black has a very long T2 and is white. And we're beginning to see differences in the gray and white matter tissue. But over here, a pretty crappy image. This is looking at the diffusion of water, and that's where we're going to spend our time today, looking at the diffusion of water. Turns out that the diffusion is a very sensitive thing to stroke. When a patient undergoes a stroke, the cells stop pumping potassium and sodium back and forth. So that part of the metabolic process stops. And water that's in the interstitial space, the space surrounding the cell, goes into the cell. The cell swells just a little bit. But the tissue, the stuff in the cell, the cytosol, the goo of the cell, has a really, it's like syrup or worse. Diffusion goes down factor of 10. So if you're measuring diffusion and you have just a little swelling in the cell, you get a big signal. So that's what people stumbled on. A guy named Mike Mosley at Stanford, really very clever guy. So how do we use this?
Wait, can you go back one slide?
Yes, sir.
So on the left side, T1, you're measuring, you excite the cells, and it's measuring the rate of decay of that.
It's measuring the rate of energy transfer out into the world.
Right. So what is different with T2? What are you measuring differently?
And this one, it's rapidly pulsing.
Okay.
I'm pulsing rapidly. When I pulse rapidly, some of the tissue has not yet recovered.
Yes.
So this is a recovery time, and this is a decay time.
Okay.
All right? He's making me honest.
He may be our next speaker.
This is from a friend of mine who went to Oxford for
It's still scary.
classics, so.
Yes, Jennifer.
So just to back up for a second, as far as the measuring proton, basically electrical activity.
You're not measuring, you're measuring the magnetic interaction.
The magnetic
Go ahead.
interaction, once that, yeah.
That's
So
correct.
why proton, like, is it the size, is it the charge of protons as opposed to, like, say, electrons or other? Oh, did you already go over this?
You can do it with electrons. My dissertation was electron spin resonance. It's just that in the human, there are very few unpaired electrons. If there were unpaired electrons, we'd all be very reactive.
Yeah.
And we're all Presbyterian.
No, no.
We're not. We don't. But protons, you have to have unpaired protons. And there are, most of the atoms in the body they have paired protons. Only water, you will find a few others, xenon and helium-3. I actually started a business to do helium-3, and people use that now.
The noble gas?
Yes.
Yeah.
But they can have unpaired spins, so
Yeah.
it's only the unpaired spins.
That makes sense. I didn't think
So here is my brain imaged about 8 or 10 years ago using this diffusion technique. Now this is a pretty crappy image because to do diffusion imaging you have to take a whole bunch of images and then do some math. So they give up on spatial resolution because you get more contrast resolution. So these are voxels that are on the order of 2 by 2 by 2. That's 8 times bigger than what you'll get if you go in for a MR scan today. Typical MR scans of the head will be 1 by 1 by 1 millimeter. But these are 8 times worse. but you are getting this contrast. So here's some detail. This is the brain stem here going down into the spinal cord. This is the cerebellum. You can see some differentiation between gray and white matter here. It'll get much better once we start doing the math. Here is our mouse, and the voxels now are 15 microns on the side. So these are 2.4 million times smaller. Each little voxel is 2.4 million of these would fit in one of these. They're long scans. This scan took 10 days. So, you know, you start the scan and go off and have a cup of coffee. We've made it much faster now. But what you can see here is again the brain stem, just like this. But now you see it going out the spinal cord. You can see the gray and white matter distinction in the spinal cord. And look, this is really quite cool. See this black line? That's the cerebellum. And that black line is a single layer of cells, Purkinje cells in the brain. So we're imaging single layers of cells. And we've taken it from 10 days to now 22 hours. So
Is that a slice or a whole brain?
No, it's a whole brain. And it's a whole brain in the skull. That's a big deal. We take the brain in the skull and scan the whole brain. So it's perfectly like the live animal in the sense that the geometry is as good as you're going to get.
But it's, the animal's dead.
The animal is dead.
Okay.
So, I mean, the backstory here, when I started commuting back and forth to Milwaukee, I would commute 2 times a month. Like Ronald Reagan's book, I was told I was president. I was told that I had children. I mean, I was in the Milwaukee, in O'Hare, many, many times. Early on, we could only make the radio frequency coils that big. So we would scan my brain in the morning, and in the afternoon, we would scan Rags the Wonder Dog, because we needed to be able to see the liver. We needed to be able to see the kidneys. The expectation was you would not be able to see them at 1.5 Tesla. So Oxford would make the magnet, a 0.3 Tesla magnet. They build a system up there. Rags and I would go up, we'd scan us, come back home. They'd build another system at 0.5 Tesla. Rags and I would go up again, come back home. 0.8, 1.3, 1.5. Finally at 1.5, we were scanning. It was the 6th of January, 1981. The Brookfield Motel, maybe 11 o'clock p.m. There's an ex-Marine who goes along to keep Rags anesthetized and make sure that he's healthy. Rags has become one of my pets. I've got 2 other dogs, Maggie and Patches. Maggie, Patches, Rags. Talk about a motley crew. Anyway, Lishko, Mike, keeps the dog well during the afternoon. We go back to the hotel, 11 o'clock. I'm sneaking him in the back door of the Brookfield. Got a dog wrapped in a blanket. I'm hoping that nobody knows. I put the dog in the bathtub because when he comes out of anesthesia, sometimes he's sick and I have to clean him up. I said, man, we ought to be able to scan a smaller animal. And that was what led me to do microscopy, because smuggling Rags in through O'Hare, out through Brookfield, Rags lived a healthy life at our house for 15 years, 16 years, something like that.
Wow.
So, back to diffusion. A thought experiment. Here's a brain, a human brain. This is something called the corpus callosum. It's a chunk of white matter. White matter is the wiring of your brain. You put a drop of India ink in the middle of that brain and wait tens of seconds. You'll see it diffuse out, and you might expect it diffuse out uniformly. It doesn't. It will diffuse out very non-uniformly because the structure of the brain is not isotropic. The diffusion is telling you about the cytoarchitecture, that is the cytoskeleton of the brain. So we use that to exploit this cartoon. This is a neuron. This is the axon that projects from the neuron. So you'll have a neuron on this side of the brain, and they will project axons 10, 15 centimeters through your brain to connect to the other side. It's the wiring. It's just glorious. These are little bundles of myelin, a protein that unfortunately in my brain is degrading rapidly. And when your myelin goes downhill, you go downhill. The conductivity goes downhill. If you measure the diffusion here, it's wonderfully isotropic. No matter which direction you look, you get the same value of diffusion. If you measure it here, because it's constrained by the axon and the myelin, you get a different number, and you get a number that's dependent upon the direction of diffusion you're trying to measure. Everybody with me? How cool is that? So here is a mouse. This is the nasal cavity orienting you. These are all the, when the mouse gets a cold, they really get a cold. They've got big nasal cavities. This is the brain. This is the back of the brain, cerebellum back here. This is all the muscle tissue. And there's a bunch of muscle up here. So now I've turned a gradient on while I'm acquiring the data in this one. So as I go from here up to here, the magnetic field changes. Still points always in the same direction, but the magnitude is changing. So the magnitude down here is lower than the magnitude down here. If something is diffusing in this direction, it will lose its signal much more rapidly than something moving in this direction, because something moving in this direction is going perpendicular to the field. So this is something, white matter called the anterior commissure, and you'll see it's kind of going black quickly there. That's because it's sort of like a bundle of celery. It's got little channels, the axons projecting into it. You see it decaying, but over on this side, the gradient is going across those axons, and the axons are keeping the diffusion from being as significant. Turn the gradient around, the same thing happens, but in the opposite direction. So now I can measure the magnitude of diffusion and the direction. So if you are a neuroscientist, you may own the King James Bible, but you will certainly own this book. This is one of the 50 most cited books in the scientific literature. It's written by a guy named George Paxinos, who is a wonderful human being. George spends half his time in Australia and half his time in Greece. He commutes back and forth. He's been to the lab half a dozen times. He comes in with a long roll of tracing paper, and he creates this book by taking glass slices of a brain, staining them with something called nissl that highlights structural, these are individual cells, and then he knows that this is the LPAG. I don't know, that could be the lady's professional alternate call. All of these structures, the map. So if you are working on the mouse brain and you want to compare your results to somebody else, you have a copy of this and you report what you find relative to this. This was published 2 years ago, and this is, I think, so this is our atlas. It was assembled as a stereotactic atlas. This is a micro-CT through the skull, primarily to get to these little sutures at the top. Those are external landmarks. So if you are doing external studies, if you're doing stimuli of a mouse, you can have a reference frame. We have colleagues over here at UNC that do functional MRI, and we have now put our MR data into that, so we have external landmarks and internal. This is isotropic spatial resolution, so no matter how I cut that tissue, I'm just spinning the axis around. No matter how I cut it, I've not lost any spatial resolution. So if I have sliced my section this way, and John Jones has sliced his this way, we can make the computer bring them together. This then has been done with diffusion imaging, where we can compute a whole bunch of different types of diffusion. Here's my Purkinje cells again. That's in something called the mean diffusivity. We do some calculation, we get a different one called the axial diffusivity. This is now just the diffusion weighted. And keep your eye on this. This region is called the hippocampus. We'll come back to it multiple times. I know the direction, so I can color code it. If something is going left, right, I can make it red. If it's in and out of the plane, I can make it blue. Back and forth, I can make it green. So I've got an enormous amount of information. I can now label it, because I got about 8 different contrasts that let me determine all of these. You get a bot, you get an AI to help you figure out what this combination means, and now you've labeled everything in 3D. We're going completely off the trail for a moment now with something that's not MR. I said you can do MR on something and then something completely different. This is stuff that was started by a guy named Carl Dieseroff at Stanford, extraordinarily bright guy. Carl will probably get a Nobel Prize at some point. He said, okay, we've got this extracellular space and an intracellular space. You stick your leg in a swimming pool and you see the altered perception of your leg being, that's index of refraction. If you can make the index of refraction between the extra and intracellular space go away, light no longer scatters, and the brain becomes absolutely transparent. You can see right through it. You build yourself something called a structured light illumination microscope, a SLIM scope. You take a sheet of light, you spread it out, and you scan it through the brain. Now, if you illuminate a chunk of tissue, almost anything, you'll get fluorescence. You can get autofluorescence, or you can get fluorescence for very specific wavelengths. You can label all sorts of things. So this is a mouse that has been created so that it puts one of these proteins called green fluorescent proteins. I'm not kidding. A guy named Roger Chen, another wonderful guy, Berkeley, Nobel Prize. Those are the individuals, those bright dots are individual cells. And these projections coming out here are the axons. These are all projections. So how cool would it be if we could make these work together? That would be really good. And you can. The problem is, once you take the brain out of the skull, it does this. Once you hand it over to the technicians, they do this. And so this is the way we get it back, really distorted, terribly distorted. But I've had a very bright graduate student who said, that's your dissertation. Figure out how to fix that. And we now get the data like this, diffeomorphically mapped. I think everybody in the world, that's the first question. What does diffeomorphic mean? You ready? You're going to ask me that, right? You can squish things together. If you've got a big enough computer, you can match anything. And so you can map these back into the MR image, so you're correcting for all this distortion. And so that's what's happened here. And you'll see, this was a most recent grant application I was trying to get funded. We did. This is one of those 3D images. This is the light sheet image merged and registered with it. You can see all of these white matter structures. You can see now this is the MR, the grayscale, and the light sheet showing us cellular attributes. And now I've added another MR contrast. At every point, I know from the MR what the direction of diffusivity is, so I can start tracking. I can start tracking the cells. This is hippocampus again, all the cells, and the dendrites, the axons, pointing toward the center. If I go out, now I'm seeing these that are derived from the MR. Those are tracks that we're seeing at 5 microns. 25 million times higher resolution than the clinical scan. These are the light sheet. These are the axons. So that's diffusion tractography. Another really clever guy, Susumu Mori at Johns Hopkins, a fabulous woodworker. Susumu has made some of the most elegant woodworking you should probably take. But he has to do all the woodwork in his kitchen because he lives in Hopkins, and he was so envious in my shop that I thought I might get him to move. So Silver said, All right, I go to this region of the image, this voxel, and I know from the MR the direction of maximum diffusivity. Mark it. Go to the next one, mark it. And so you can start laying out the tracks, the wiring diagram of the brain. But there was some more work. The problem is, the Lord has not made it simple. You think that there's one set of tracks going through every voxel. There aren't. So there would be some coming from this direction, some coming from this direction. How do you distinguish those? If you're tracking in this direction and you get into a voxel and there's one going off in this direction, you keep track of that. That's a tough problem. Turns out that this young man, Evan Calabrese, I think is the smartest graduate student I've ever had. It's a toss-up. I've had a couple pretty good ones. MD, PhD at Duke. And this is some of his work. This is a single slice from one of the representative images. This was a 10-day scan. We now do them in 22 hours, but this was 2015. Single slice. This is the calculated color index image where we know what's going forward, what's going left, what's going right. And from the math that I'm not going to bore you with, I couldn't reproduce it, you can go in with enough measurements and you can say, all right, I might have two types of fibers in that. Let me unwind them. So you unwind them and you can get a red fiber population and a green fiber population. When you do that and you map against the atlas, you can come up with a region of interest, the hippocampus, and then you go to that region of interest and you go in the middle and say, put a seed right there and tell me where it takes you. And this is the hippocampus connected to the rest of the brain. The hippocampus is connecting over to the other side, the contralateral side, but on the same side it's connected much more chromatically. We're back to our atlas where we've got all these labels. These are the labels that everybody is using now. So if you want to know where Chapel Hill is, you label it Chapel Hill. If you want to know where motor cortex is, you label it in this atlas motor cortex. This is the skull, still in place. Oh, look at the ear. That was the cochlea of the ear that just went by. Jesus. Yeah, it's fabulous.
What is the current limit? So, you know, you were showing your graduate students' work where it was saying if you have sort of two axials, one going this way, one going this way in the same space, you can untangle them and show A and B. Can you also show C, D, E, F, G,
You
etc.?
have hit the crucial question we're fighting right now. There are a couple of papers out there, one of which a guy named Meyer Heim, a German guy, with 78 authors on it. I think if I put everybody in here on it, I could create a paper with 70. I don't know 78 people. And they looked at human imagery, and they found out that in tracking, they had more false connections than true connections by a factor of 4. So in science, when you got 4 times as many wrong answers as right answers, it's a very controversial thing. But remember, what we're looking at are voxels that are many, many thousands, even millions of times smaller. So we're down to the point where we have, the voxels are such that we have maybe 10 or 12 fibers. Mm-hmm. And when we do the math the way we're doing it, we can resolve 3 or 4 populations. And for most things, that seems to be sufficient. We still should be humble. We should be humble because we don't get it right all the time.
And you're saying like fibers. A fiber is how wide?
A micron or 2.
Okay.
I mean,
A micron or 2.
The fiber, that's another interesting question. Axons come out and they bundle together to create a fascicle. Fascicles are easy to check. The individual axons are a little bit harder. The individual axons are on the order of micron. The fascicles can be on the order of a couple of microns, so it's easier. But we're still not perfect, but that's a good thing. Being not perfect is always a good thing.
How do you isolate the voxels?
Pardon?
How do you isolate the voxels, each voxel that you study, that you're trying to to differentiate.
You look at everybody, you have a ginormous computer that looks at all of them at the same time. I have more compute power than you can imagine. I have one computer that has 1,000 processors in it, and we'll see that in a minute. I'll get to that question. So you can do this sort of thing. Cortical spinal tract is a very important part of the spinal the neural mechanism. And you can say, tell me everything that's connected from here going out the back of the spine. Look at where the part of the foot connects up here. So you can look at individual circuits. So this is fine. I'm sorry it's taking longer than I should. I'll move ahead. Can you do anything useful with this? Is it just abstraction? It's abstract. It's cool. It's cool. It literally keeps me up at night. I think of these images and I think, wow, I can figure it out. So we're looking at that in Alzheimer's disease. The problem with Alzheimer's disease is genetics. If you try to do a genetic study, the typical thing is called a genome-wide associative study. And unless your entire population is twins, you have no control over it. You have whoever walks in the door. They could be 35, they could be 38. You could filter so that you have only those who are 35 and 38. But if you're looking at patients with Alzheimer's disease, find me patients with 35 and 38 who have Alzheimer's disease, or that don't smoke, that don't have the genes that are going to predict Alzheimer's. Controlling for this is just a nightmare. So a typical GWAS study has to have at least 50,000 patients to get statistically significant done. So what can you do about that? You can control the genetics. So we're working with a very clever colleague, friend for 10 years now at the University of Tennessee. And he's created the BXD population. Most of the population study, most of the studies with mice will look at the B6 mouse. The B6 is the standard mouse that they look at all the time. This drives Rob crazy. He says if you've looked at the B6 mouse, you've looked at N of 1. So maybe your B6 mouse has a response, but another mouse that has a different genome will have a completely different response. So what Rob has done is created the BXD, the B6 crossed to the D2. They cross, back cross, back cross, and do the Mendelian things, the same thing that Mendel did, and create this population of animals with BXD29. So he's got a whole bunch of BXD29s. And if you look at one BXD29, its genome is identical to the next one. So now you can start doing replicates. You don't have to have two boozers to scan. I can look at Carlos Boozer, Dad, I can look at any boozer. You don't have to go out and recruit twins. You've got a population of boozers. Gosh, if we have a population of boozers. I just gotta get Rob working on that, it's a population of boozers. So he's got this dialed-in collection of animals that have the broad diversity of this population here, except we know exactly what it is and we can replicate it. So now we can do a study with 40 different strains, and we can do it with about 500 animals, which we've just completed after 6 years of study. That's just been published. Here is the workflow. I have a magnet that's 9.4 Tesla. It's a pretty small magnet. It's about this big, but it's very high field. I can't scan anything bigger than about that, but I can do a really good job scanning that. I put into it a set of braiding coils that were made especially for us by a company, friends up in Cambridge, that are 1,000 times stronger than what's in a clinical magnet. So I can bang it like crazy. And I started up, and I have this computer infrastructure with something called a cluster. Each cluster has 20 or 30 nodes. Each node has 20, 30, 40 different processors. And so I can say, computer, just keep things running. Scan me one scan, then do the scan. I can go off and have a cup of coffee. I can go home. I can have dinner. I can go to bed. Come back the next day, and it's gathered 120 volumes. That all gets automated. The next thing that happens is we have to process it like crazy. It turns out I get one image like this, and I need the next one. to be right on top of. But because I'm putting such great gradients on it, it might be moved by 10, 15, 20 microns. So I have to have a computer redo that. So we do all of that in automated pipelines, push it through our atlas, and now we add the light sheet to it. We then take a group of animals, let's say 12 BXD29s at 6 months, 12 BXD29s at 6 months, with a transgene that causes Alzheimer's. And now I can start pushing those through and create a population atlas, which spits out a whole bunch of spreadsheets. I was talking to Chris about the amount of data that Glaxo might undertake for a clinical study. And they're talking about something on the order of megabytes, something. We're talking about petabytes. And for every animal, we get, this is a single slice from a 3D. This is the diffusion rate of AD. I have 12 different contrasts. If you look, this boundary right here, I can see pretty well there. I don't see it well at all over here. This structure right here is called the red nucleus. I can see the boundary very well there. Can't see it at all down here. But if I look at all of them and I get the computer to survey what's going on, I can outline 230 different regions of interest. Extraordinarily precisely, within 2%. So when I do that, then I can go into the computer and say, let's look at the hippocampus, for example, break it down. These are 4 of the most critical parts of the hippocampus. Tell me exactly how big they are. Tell me how long they are. Give me a shape analysis. Give me all the details that you can for the BXD29, for the BX29 with the Alzheimer's disease, for BXD43, etc. And you just leave the computer working away. So this was published last week in Nature Neuroscience. Nature is one of the Gucci journals. You have to walk on water to get to it. They're extraordinarily picky. But we were able to convince them that we could see changes that were related to Alzheimer's. This is the light sheet. You've all heard about A beta, the compound that accompanies Alzheimer's, which is thought to be the source of the breakdown. So this is the light sheet 3D. This is the MR 3D. Here is a 2D version of it, registered together. We can zoom in. At this spatial resolution, we can say, tell me what the percentage of that area or that volume is, A beta. Go over to the new N image. Tell me how many neurons I have in the subiculum, how many neurons I have in CA1, and then look across all of the strains and tell me what's changing with and without Alzheimer's disease. We thought and we postulated that there might be genes that would be resilient. We all know there are plenty of people that have the Alzheimer's disease genes, and they're fine. So why is that? Why can one person get AD, they've got the gene, and the next person has the same gene, but they're fine? There's got to be a resilience gene. And this demonstrates that. This is the BXD32. This is another one of those things that keeps me up at night. We've just created a population atlas. We've mapped them all together, and we've said, tell me what the percent difference is for just one of these 12 different metrics that I have. And I can see the BXD32 is pretty varied, BXD65 pretty varied, but BXD77, everything is going downhill. And it turns out, from a behavioral standpoint, that's one that is the most effective. So the last thing is tracks. This is the hippocampus again. And now I've seeded it and said, tell me what the wiring diagram looks like. So here's the normal wiring diagram, connected over to the ipsilateral side. Here's the breakdown, and I can take all of these wirings and do some fancy math to give you a measure of the connectivity. So we put all of that together, and now we can do these studies. We're doing them for Huntington's disease, Parkinson's disease, any neurodegenerative disease. And
So you can identify what's happening, but you can't identify why it's happening.
maybe why. If we know, for example, I haven't shown you the whole story of the light sheet. With the light sheet imaging, I have hundreds of different molecular markers that I can use. So I can look at any marker having to do with any pathway that might be altered. And I can decide whether that marker has been changed in the hippocampus, in motor cortex, in senescence, so metasensory cortex. So it's dependent on knowing something, some prior about the disease. But once you do, there's a lot you can do. So, surprise, surprise, I don't do much of any of the work. I go into the office, make myself a cup of coffee, sit back, it all happens. That's the first image, one of the first images I took at Duke in 1974. It was the second EMI scanner in the United States. The first one was at Mayo, the second one was here at Duke. And that's what an image looked like. It took 38 minutes to take that one image. Here's the eye candy, sum of the Lord's best work. This is a track density image at 5 microns through a mouse brain, color-coded. We're going from the nose through the hippocampus. It's color-coded like I mentioned earlier, all the way through. I can zoom in on these. The screen here is not big enough to accommodate this. So with that, I'm finally done.
Well, my question really deals with something that's completely off from what you're presenting. And that is,
I should go right.
well, the question really deals with the fact that oatmeal is thought to be anti-inflammatory, protects the brain, et cetera, They postulate that various components within oatmeal does this, but
I find that mixing a third of a cup of oatmeal and a third of a cup of
Brown sugar.
barley. So that's my recipe. You're asking, can this be relevant to that?
yes.
Look, I'm an engineer. I'm not a neuroscientist. I'm not a molecular biologist. I don't know anything about the disease. What we've got is a platform for studying things. So, for example, we are looking at several studies. We will be doing studies where we're looking at dietary interventions. So we will be able to give a quantitative metric of whether that brain has changed because you keep one population on the diet and one population off the diet. So yes, those sorts of things are possible. We have a grant pending at NIH at the moment that will be reviewed week after next. We'll know whether we are past the Trump barriers or not. And some of those data sets, the animals that have been kept on these different dietary, rapamycin is something that is thought to be particularly relevant to AD. And we've got animals that have been maintained on rapamycin.
What is rapamycin? Is that an anti-inflammatory?
It is something like that. It's one of the weirdest things. It was found in a fungus on Easter Island. It's one of those things. Where did they come up with that? But yes, rapamycin is a rage drug right now that people think might be effective. So yes, we have a platform that could be useful for that. How sensitive is the platform? It's certainly a lot more sensitive than anything else people have. So, will it be sensitive about? Don't know, but we think so. We think that this platform will become a standardized platform for doing all sorts of neurodegenerative studies. I mean, look, you can say one set of animals, dial up the genetics you need, do the exposure, make them smoke cigarette, make them eat oatmeal, whatever. It's a simple experiment, Kathy.
So what's next? Have y'all figured out every detail of all these, slices, microns, and all the little color codes that you want, or there are more to come?
I'm 79.
So there's room for more stuff to.
There's tons of room. I mean, as I said, it's a platform that can be applied to any one of a number of diseases. We just presented a paper out at Alzheimer's disease, California, on Huntington's disease. And we can see the evolution of Huntington's disease, and they are looking at something called the CHDI Foundation is funding that. And they have a couple of compounds they think might diminish the consequences of Huntington's. It's a genetic defect, but they think they can modulate that effect. So we're looking at that right now. Parkinson's, something we could look at. Just did a study with NIEHS looking at chlorpyrifos, which is a nasty toxic chemical. Everything that you do bad for the brain is a candidate for us to study. Yes, I'm back.
Yeah. Neurosciences, but I know that the beta amyloid stuff has been what you kind of get most on the receiving end in the press on neurosciences with respect to Alzheimer's. Are you able to measure beta amyloid presence with these scans?
Yes.
I figure since it's a protein, protons might be present, you know. So yes to that. The next
So
question.
the slide that had all the yellow, that was light sheet,
Yeah.
which is staining for A beta.
A beta is beta amyloid, I figured, yeah. And then the next.
And then we not only measure, we get a very quantitative metric because our resolution is so high, we can say, go in there and count the volume of this structure that is normal and count the amount of A beta. So you get a ratio of that, you get 43.6% of that
Yeah.
structure is full of A beta. So
And
you
is
get a
there-
very accurate measure.
That's really cool.
Well,
And
It's really.
yeah,
You get the collection of the beta before you see the pruning of the neurons. Which comes first?
So it's the sort of question we're able to ask, we've not asked. So what I showed you was one time point, 14 months. We showed you with animals with AD genes and without at multiple strains. And we can show some strains do, some strains don't, with and without. The next thing to do is to look at 6 months. We have animals at 6 months, we've scanned it at 6 months, but there's an enormous amount of data to digest here. So digesting that and say, did we see it at 6 months and 14 months?
Because the change may be some 1 year.
Yeah. And we just have to dive through and dig out the data. Once you acquire the data, that's 10% of the business. 90% is reducing it to something you can get your head around. And really, truly, literally, that is my problem right now. I'm losing it with age, and I can't get my head around it. Yes, sir.
Is it dangerous at all or harmful to take an MRI?
No.
No?
No, I scanned my children multiple times when we were first starting. I've been scanned multiple times, no. My dissertation is actually on the mechanisms of radiation damage, and it turns out that the amount of energy in the radio frequency pulse that you put in is vanishingly small relative to the amount of energy in an X-ray. X-rays are energetic enough that they can break molecular bonds. And there is no way that the energy in a radio frequency pulse can break a molecular bond. So from that standpoint, no. The danger in MRI comes from the fact the MR system is always on. So we've had a number of cases at Duke and around. Nurse is trying to help. Middle of the night, they're scanning a pediatric patient. The tech is working to get the patient in and out. The nurse wants to help. She brings the oxygen bottle in. Oxygen bottles are steel. Oxygen bottle goes out, goes soaring into the magnet, hits the nerve, hits the tech, shatters her arm, but the child was fine. I can show you, I don't have any, I can show you mop buckets stuck in my magnet. I can show you a motor. Graduate student, I told you, don't take anything close to the magnet. Well, I didn't think it would
What about a lipstick was in our pocket?
So,
For our hard ones, and it went through.
so the biggest, when this first scanner came in, I ran it for the first 6, 8 months. We had no technologists. I mean, who would run it? I was the guy running it. And I would give constant demos to everybody. And everybody would come in and I'd ask them all the questions, you know, you don't have a pacemaker. Pacemakers will shut down, that's not good. Hearing aids, I can tell when I'm too close to the magnet, my hearing aids shut down. But I would notice women come in and they would begin doing things now. Anybody with an underwire bra? Somebody had an underwire. So there are hazards to MRI. They're all mechanical. But there is you can, as you go to higher and higher magnetic fields. Those systems at Duke are 1.5 and 3 Tesla. UNC has a 7 Tesla. And when you get to the higher fields, as the radio frequency goes in, it's harder to get in. It deposits more energy. And so you can get to the point where you can heat. And so you can cause heating, and the scanners all have protections on it to keep that. So there are some heating hazards. There's a guy.
What does it heat?
Pardon?
What does it heat?
It's microwave. It's a gigantic microwave.
I mean, I'm thinking about the rods and screws in my back. Is it going to heat those?
You cannot go in this scanner. You will not go in the MR scanner again. The hazard for you is something called eddy currents. If you have a chunk of wire, when you turn the gradient on, you're changing the magnetic field. And one of Maxwell's equations says that when you change the magnetic field, you can cause a force. That's the way electric motors work. And it'll generate a current in that chunk of, for the earliest scans, I was scanning a number of women, and I kept coming up with these women that had gigantic holes in the brain. I said, you know, my wife's a lot smarter than me. I know she doesn't have holes in the brain. Anybody want to guess the reason? Eyeliner. Eyeliner has metallic particles in it, and that'll generate a little current in the eyeliner.
Tattoos too?
Tattoos.
I'll have to remember that.
Some tattoos. So there are some hazards there, and they are monitored very carefully.
I heard a lecturer one time talking about something similar, and he mentioned the dangers of it, and you shouldn't do it very often. He goes, but I get one about every 30 days because they're free, because I work for the hospital.
So I would have no qualms, again, I had no qualms about sticking my sons into the magnet. I would take them in on weekends.
You didn't mention clinical, but I'm assuming that they use the MRI just like they do for the heart to map the areas where they're going to ablate. Would they be doing that for Alzheimer's?
We don't ablate with Alzheimer's.
Or other issues with the brain? Do you use mapping to fix them?
You're asking a complicated question. For cardiac ablation, that's usually done with electrical mapping. And the standard way is to have a sock that's got a whole bunch of electrical contacts in it. And you open the patient up, and you put that sock around the heart, and then you map all the electrical activity into a computer 3D map. So that mapping for ablation is frequently done without, imaging may be done. The first ones that were done, I did imaging in CT. But you would not do cardiac ablation mapping with MR for the same reason that you could generate currents. So if you've got a sock or something conducted in there, you don't want to be turning those gradients on. Brandy?
Just think about the size. A lot of your work's in rats and little small things. Are there years away, or is it pretty soon that there's an application to doing studies in humans?
So I get asked that question all the time. When are you going to make this clinical? I don't mean this to be caustic, but I don't care. I'm interested in the basic sciences. And I can do things in the basic sciences that I care about.
Yeah.
There are some, will the spatial resolution improve in clinical? Yes, all the time. I have a colleague at Duke who works in BIAC, the Brain Image Analysis Center, and they have been pushing the spatial resolution, and they can get diffusion tensor images at about 0.8 millimeters, 800 microns. I'm at 15 microns. So it's apples and kumquats. Yes, sir.
On the engineering question, what's the biggest constraint on your fidelity as you try and get more precise imaging? Is it magnetic strength?
In real estate, it's location. In MR, it's signal. So if the voxel is 2 million times smaller than the clinical machine, there's 2 million times fewer protons to sample. Your signal is 2 million times weaker. So you can get some of that signal back by going to higher fields. So if I go from 1.5 to 3 Tesla, I'll get a factor of 2. A factor of 2 when I'm down a factor of 1 million doesn't help me a lot. So you got to do a whole bunch of things. That's why clinically, you're not going to see tens of microns. You will see in some situations, I see NIH grants that come through where people, there's a scanner up at Harvard called the Connectome scanner. And it's a 7 Tesla system. And they have ginormous gradients. Literally, they have their own substation. The amount of power going into that substation, whenever they give a lecture, my colleague up there gives a lecture and he puts out a Trident-class nuclear submarine, has a picture that says, we use the same amount of power as is generated in the nuclear submarine on the slide on the right. Enormous amounts of power. And they can get to 500 microns, I think. So the biggest thing that we have going from it, if you have to excite a volume that's this big, or you have to excite a volume that's this big, the difference between that and that goes as 1 over the radius to the 6th power. So it's really, really hard to get the spatial resolution down below, for diffusion tensor, down below even a millimeter. There are some things that we'll see down maybe to 300 microns, something like that. But that's orders of magnitude from where we are. I'm just losing.
Are all of your grants or most of your grants from the government? Does anybody else give grants?
Yes. You did bring in a basket? What? There's no basket. So I have a foundational CHDI Foundation that studies Huntington's disease. I have some industrial grants, so I've had funding from Pfizer, the largest. Duke is just, for years, in about 83, 82, I established the laboratory as a national resource. And it was the longest running national resource, I think still to this day, 37 years I ran it. And there I could get outside support from any industry that wanted to use the special resources. Duke always fought it. Last week, we got a missive saying, wow, that was a really good idea, we should do that.
37 years later.
37 years later.
I'm getting around there.
So funding is hard. Funding is
Is the
really
funding
hard.
all from grants that you write though? Is it you have to keep it up?
All the time.
Yeah.
So much of your time
Somebody I know worked for you.
How much of my
spent doing grants versus
salary.
Oh, goodness. I could be glib and say all of my time, but it's kind of, I would say 70% of my time is idea and implementation of grants or monitoring grants or doing that thing. But I'm surrounded by it. I can't do any real work anymore. I mean, I'm totally useless in the lab. I sometimes have decent ideas. It turns out that walking the dog is a really good thing to do. Periodically, I'll have a decent idea, and the dog gets a treat. But the people in the lab are the ones that are doing the real work. So I make coffee. Thank you all for taking the time to listen to me.
Thank you so much.